Let's talking about parasitology today. Parasitology is the study of parasites, their hosts, and the relationship between them. As a biological discipline, the scope of parasitology is not determined by the organism or environment in question but by their way of life. Parasites occur in two distinct forms. Single-celled protozoa. Multicellular metazoa called helminths or worms. Protozoa, also protozoan, plural protozoans, is an informal term for a group of single-celled eukaryotes, either free-living or parasitic, which feed on organic matter such as other microorganisms or organic tissues and debris. Historically, protozoans were regarded as one-celled animals, because they often possess animal-like behaviors, such as motility and predation, and lack a cell wall, as found in plants and many algae. Although the traditional practice of grouping protozoa with animals is no longer considered valid, the term continues to be used in a loose way to describe single-celled protists, that is, eukaryotes that aren't animals, plants, or fungi, that feed by heterotrophy. A number of protozoan pathogens are human parasites, causing diseases such as malaria, Plasmodium, amoebiasis, giardiasis, toxoplasmosis, cryptosporidiosis, trichomoniasis, Chagas disease, leishmaniasis, African trypanosomiasis, sleeping sickness, acanthamoeba keratitis, and primary amoebic meningoencephalitis, naegleria. Parasitic worms, also known as helminths, are large macroparasites. Adults can generally be seen with the naked eye. Many are intestinal worms that are soil-transmitted and infect the gastrointestinal tract. Other parasitic worms such as schistosomes reside in blood vessels. Parasitic worms live in and feed in living hosts. They receive nourishment and protection while disrupting their host's ability to absorb nutrients. This can cause weakness and disease in the host. Parasitic worms cannot reproduce entirely within their host's body. They have a life cycle that includes some stages that need to take place outside of the host. Helminths are able to survive in their mammalian hosts for many years, due to their ability to manipulate the host's immune response by secreting immunomodulatory products. All parasitic worms produce eggs during reproduction. These eggs have a strong shell that protects them against a range of environmental conditions. The eggs can therefore survive in the environment for many months or years. Many of the worms referred to as helminths are intestinal parasites. An infection by a helminth is known as helminthiasis, helminth infection, or intestinal worm infection. There is a naming convention which applies to all helminths. The ending asis is added at the end of the name of the worm to denote the infection with that particular worm. For example, Ascaris is the name of a type of helminth, and Ascariasis is the name of the infection caused by that helminth. Entamoeba Histolytica is an anaerobic parasitic amoebozoan, part of the genus Entamoeba, predominantly infecting humans and other primates causing amoebiasis. Entamoeba Histolytica is estimated to infect about 35 to 50 million people worldwide. The word histolysis literally means disintegration and dissolution of organic tissues. Transmission. The active, trophozoite, stage exists only in the host and in fresh loose feces. Cysts survive outside the host in water, in soils, and on foods, especially under moist conditions on the latter. The infection can occur when a person puts anything into their mouth that has touched the feces of a person who is infected with Entamoeba histolytica, swallows something, such as water or food, that is contaminated with Entamoeba histolytica, or swallows Entamoeba histolytica cysts, eggs, picked up from contaminated surfaces or fingers. The cysts are readily killed by heat and by freezing temperatures, and survive for only a few months outside of the host. When cysts are swallowed, they cause infections by existing, releasing the trophozoite stage, in the digestive tract. Symptoms can include fulminating dysentery, bloody diarrhea, weight loss, fatigue, abdominal pain, and amoeboma. The amoeba can actually bore into the intestinal wall, causing lesions and intestinal symptoms, and it may reach the bloodstream. From there, it can reach different vital organs of the human body, usually the liver, but sometimes the lungs, brain, spleen, etc. A common outcome of this invasion of tissues is a liver abscess, which can be fatal if untreated. Ingested red blood cells are sometimes seen in the amoeba cell cytoplasm. Pathology. In the vast majority of cases, infection is asymptomatic and the carrier is unaware they are infected. However, in an estimated 10% of cases Entamoeba histolytica causes disease. Once the trophozoites are excisted in the terminal ileum region, they colonize the large bowel, remaining on the surface of the mucus layer and feeding on bacteria and food particles. Occasionally, and in response to unknown stimuli, trophozoites move through the mucus layer where they come in contact with the epithelial cell layer and start the pathological process. Entamoeba histolytica has a lectin that binds to galactose and N-acetylgalactosamine sugars on the surface of the epithelial cells. The lectin normally is used to bind bacteria for ingestion. The parasite has several enzymes such as pore-forming proteins, lipases, and cysteine proteases, which are normally used to digest bacteria in food vacuoles but which can cause lysis of the epithelial cells by inducing cellular necrosis and apoptosis when the trophozoite comes in contact with them and binds via the lectin. Enzymes released allow penetration into intestinal wall and blood vessels, sometimes onto liver and other organs. The trophozoites will then ingest these dead cells. This damage to the epithelial cell layer attracts human immune cells and these in turn can be lyssed by the trophozoite, which releases the immune cell's own lytic enzymes into the surrounding tissue, creating a type of chain reaction and leading to tissue destruction. This destruction manifests itself in the form of an ulcer in the tissue, typically described as flask-shaped because of its appearance in transverse section. This tissue destruction can also involve blood vessels leading to bloody diarrhea, amoebic dysentery. Occasionally, trophozoites enter the bloodstream where they are transported typically to the liver via the portal system. In the liver a similar pathological sequence ensues, leading to amoebic liver abscesses. The trophozoites can also end up in other organs, sometimes via the bloodstream, sometimes via liver abscess rupture or fistulas. In all locations, similar pathology can occur. Pathogenesis. Entamoeba histolytica causes tissue destruction which leads to clinical disease. Entamoeba histolytica, induced tissue damage by three main events: direct host cell death, inflammation, and parasite invasion. Diagnosis. Diagnosis is confirmed by microscopic examination for trophozoites or cysts in fresh or suitably preserved fecal specimens, smears of aspirates or scrapings obtained by proctoscopy, and aspirates of abscesses or other tissue specimen. A blood test is also available but is only recommended when a healthcare provider believes the infection may have spread beyond the intestine, gut, to some other organ of the body, such as the liver. However, this blood test may not be helpful in diagnosing current illness because the test can be positive if the patient has had amoebiasis in the past, even if they are not infected at present. Stool antigen detection and PCR are available for diagnosis, and are more sensitive and specific than microscopy. Treatment. There are a number of effective medications. Generally, several antibiotics are available to treat Entamoeba histolytica. The infected individual will be treated with only one antibiotic if the Entamoeba histolytica infection has not made the person sick, and most likely be prescribed with 2 antibiotics if the person has been feeling sick. Otherwise, below are other options for treatments. Intestinal infection, usually nitroimidazole derivatives, such as metronidazole, are used because they are highly effective against the trophozoite form of the amoeba. Since they have little effect on amoeba cysts, usually this treatment is followed by an agent, such as paromomycin or diloxanide furoate, that acts on the organism in the lumen. Liver abscess. In addition to targeting organisms in solid tissue, primarily with drugs like metronidazole and chloroquine, treatment of liver abscess must include agents that act in the lumen of the intestine, as in the preceding paragraph, to avoid re-invasion. Surgical drainage is usually not necessary except when rupture is imminent. People without symptoms. For people without symptoms, otherwise known as carriers, with no symptoms, non-endemic areas should be treated by paromomycin, and other treatments include diloxanide furoate and iodoquinol. There have been problems with the use of iodoquinol and iodochlorhydroxyquin, so their use is not recommended. Diloxanide furoate can also be used by mildly symptomatic persons who are just passing cysts. Thanks for watching. If you like this video, please thumb up and leave a message. You must be my biggest motivation to make the video. Please subscribe to the channel, where you'll find more and more medical videos here later. Thanks.